Okay, thank you very much for that kind introduction. Before I begin, I want to start by thanking Materials today and the event organisers for the very kind introduction and invitation to present this lecture today. And in the spirit of the Materials in Society lecture, what I wanted to do was talk to you about some of the opportunities that's been brought about by diamond quantum probes. And in particular, what I want to focus today's talk on is their application for biosensing and imaging applications. And so today I'm going to be talking about some of the work we've been doing at the University of Melbourne in particular, about applying these diamond quantum probes to interesting biological systems such as primary cortical neurons. And I'll tell you about this little guy. This is a sea mollusk chitin, and I'll tell you a bit more as we go on. Before I start, I wanted to thank and acknowledge the group at the University of Melbourne. So our group is led by Professor Lloyd Hollenberg, and we're fortunate enough to have a great team of postdocs and graduate students, as well as some fantastic collaborators both nationally and internationally. Many of those are in the audience today. So I was going to start off by talking about defect centres in diamond, and I think I'm preaching to the converted in this audience here. But what I wanted to do was just highlight some of the aspects of a particular defect centre in diamond in particular. And the defect centre I'm going to be talking about today is called the nitrogen vacancy centre. And this centre is formed when you have a substitutional nitrogen atom, an adjacent vacancy, and this defect can lie in one of four possible orientations in this diamond lattice. Now this is an optically active defect centre, so if you shine light on it, so if I shine light like this green laser pointer, I'll get strong red fluorescence, broad band. The defect centre can have two charged states, so there's a neutral charged state and a negatively charged state, and it's the negatively charged state which I'm going to be talking about today. That's the one that's got all of these amazing quantum properties. So in terms of biological applications, people really got interested in this material mainly from the point of view that the fluorescence from these defects is completely photostable. And so if you compare it to other traditional bioprobes such as organic dyes, fluorescent proteins, quantum dots, the traditional probes tended to blink or bleach over time, whereas diamond is completely photostable. So that was the first real interest from a biological perspective. And Wan-Chung Cheng's group in Taiwan were able to show and use this long-term photostability to do high-resolution three-dimensional tracking inside HeLa cells. And since then, the field's really expanded in this area. We've heard a lot about, at this meeting in particular, the biocompatibility of diamonds, so this is also another important property. We can also tune the size of the diamonds that we work with, so we can work with nanodiamonds as small as 5 nanometres, all the way up to single crystal diamonds, which are several millimetres by several millimetres in size. And so today what I want to do is point out two applications that we've targeted, one with nanodiamonds and one with single crystal diamond, to give you a flavour of what we can actually do with these sorts of materials. Finally, the other nice aspect about diamond is that the surface is suitable for functionalisation. So if you want to use it as a biological probe, you'd like to be able to target it to specific cellular components in particular. And there are a range of functionalisation strategies which have been pioneered by Anke Kruger, Peter Sliger and others. And what we've found recently is these functionalisation effects and surface termination effects actually change the quantum properties of the probe. And I think this is going to be a really interesting area in the upcoming years. I think we can improve the quantum properties of these systems a lot by understanding the surface and actually using that to improve the quantum sensing opportunities. So quantum sensing with NV centres. So what actually is it? What do we do? So I'm a physicist by training, and my main interest in this material came about because this system represents an ideal quantum system that we can play with at room temperature. And so if we look at the ground state of the NV centre, it's basically a two-level system. We've got a ms equals 0 spin state here. This system is bright, so when the NV is in this state, we get very strong fluorescence. And then we've got plus and minus 1 spin states up here, and when the NV are in those states, we get weaker fluorescence. Okay, and the nice thing about this defect is that if we shine green light onto it, we can polarise the NV spin into this ms equals 0 state, into this bright state. And that's a really unique feature in diamond as a material. So diamond is one of the very few materials that you can simply shine light and polarise an electron spin. So this is a really unique aspect. So we can prepare, if you like, a quantum state just by shining light on this system. We can read out the quantum state just by looking at the light that we're getting back. So the red fluorescence that comes back, if it's strong, we know it's in the ms equals 0 state. If it's weak, we know it's in the plus or the minus 1 state. So we also have an optical readout of this sensor as well. And so what we're trying to do in our group is actually use this delicate quantum state to probe interesting environments. And so the environments that we want to target are going to act to collapse the quantum state in different ways. So there might be parts of the environment here which will collapse the quantum state very quickly, and then there's other parts of the environment that might be more forgiving on the quantum state. So we want to monitor these quantum states, and that will tell us something about the environment. That's the idea of quantum sensing with these defect centers in diamond. And so what can you do? So people have showed that using the NV center, we can detect the magnetic field from a single electron spin, which is attached to a protein, for example. Others are detecting magnetic fields from nuclear spins, and so there's a whole field now dedicated to nanoscale and NMR with this sort of system. And there's going to be a magnetometry session tomorrow morning, which you'll hear a lot more about that. Others have done electric field sensing, drug delivery, pH sensing, and pressure sensing. So all of these modalities are possible with just this single quantum system. And we can tune this quantum system to address any one of these criteria that we like. So it's a very versatile system to probe interesting properties in biology. And so I thought today I'd go over one of the simplest quantum sensing protocols that we use, which is called optically detected magnetic resonance. And so the idea here is that if we take this is the NV ground state again, we've got our 0 and our plus or minus 1 levels. If you apply a magnetic field to this defect, what you do is you Zeeman split these plus and the minus 1 levels. And what we'd like to be able to do is actually read out where those energy levels are. And that is done simply by collecting the light that comes back, as I mentioned before. So all we need to do in this system is monitor the intensity of the light coming back from the NV, and we can do this in a confocal microscope or a wide field configuration. We might have some RF line or microwave resonator nearby, which provides the energy required to flip the NV from this state into the plus or the minus 1 state. And when we get that energy just right, and it transitions, we get a reduction in the fluorescence intensity. So if we scan the microwave frequency, when we get the frequency right, we get a decrease in the fluorescence from the NV, and that provides us a direct readout of where these energy levels are. And so these energy levels split according to the gyromagnetic ratio of the NV center, and the gyromagnetic ratio is 2.8 megahertz per Gauss of applied magnetic field. And because these lines are very narrow, so these lines can be kilohertz, this provides you with a really sensitive magnetometer, and that's been the main interest. That's why we can detect electron and nuclear spins with this material. So if you want to use it as a magnetometer, in a low magnetic field, this is what a NODMR spectrum would look like. In a high magnetic field, this is what it looks like. So these levels simply split. And if we look at the amount of splitting, we can work out the strength of the applied field along the NV axis. And what's nice about diamond as a material is that the NV can actually sit in 4 different configurations. And so if you measure from each of these configurations, then you can get the projection of that magnetic field in 4 different directions, and you can reconstruct the full 3D vector magnetic field. And that's what we'll do. I'll show you some examples of that later in the talk. So that's one of the attractive parts in terms of magnetometry with the NV. The other thing that's interesting from a bio perspective is measuring temperature. So temperature has a different effect on this system. So if you monitor the ODMR spectrum, this might be what you get at room temperature. If you increase the temperature, you get a translation of these peaks. So we don't get a splitting anymore, we just get a shift in these peaks. And so that gives us a way to decouple temperature effects from magnetic field effects, for example. So these are the 2 common modalities that we use. And our group has been active in developing applications based on nanodiamonds. So we started off in 2011 getting nanodiamonds into HeLa cells, identifying nanodiamonds with single NV centers, and then using this ODMR to tell us something about the rotation of the particles in these HeLa cells. We then moved on to sensing in artificial cell membranes. So we label these membranes with spin labels, such as gadolinium, for example, and we have sensitivity down to a single spin label. And we're developing imaging platforms for these nanodiamonds. So we've developed a combined AFM confocal microscope, and we can use this microscope to image the magnetic properties of iron nanoparticles, or we can image the temperature profile of single gold nanoparticles, for example. So it's a very powerful tool. So these are the sorts of examples of what we do with the nanodiamond. And there's a different imaging modality that we're starting to develop for the single crystal diamond. And this gives us an opportunity to build, if you like, a new type of microscope, what we call a magnetic microscope. And so the idea here is that you take a single crystal diamond, several millimetres by several millimetres, you do an implant of nitrogen into the surface, and you have a 2D layer of NVs that you create at that surface. And then using a camera, we can image the fluorescence that comes from those NVs, and we can actually start to image then magnetic properties of things that you put on top of that diamond chip. So we started off with the proof of principle experiment looking at magnetic domains of hard drives from thin ferromagnetic materials. And more recently, using ODMR, we've been able to image the current flowing from graphene, which is deposited on our diamond substrate. So we can actually watch how the current flows in 2 dimensions through the graphene, and we can see defects and changes in that profile. And in addition to that, using a slightly different quantum sensing protocol, you can turn the NV into an EPR spectrometer. So this EPR spectrometer will measure the fluctuating magnetic field from paramagnetic molecules, for example. And we've used that to measure copper 2+ molecules in solution. And it's not just an EPR spectrometer like you would typically use in biology, but we can do EPR imaging. And so that's where the real power of this technology comes in. So we can do high-resolution EPR imaging, identify species of interest via the spectroscopy through the NV centre. So these are sort of the applications that we're doing with nanodiamond and single crystal diamond. And so what I wanted to talk to you today about was some of the other sensing capabilities that we've done recently. So the first one is intracellular thermometry. So this is a promising field that people are getting into at the moment. And this work was really motivated by 2 key questions. And so the first question was, does the presence of this material, does the presence of nanodiamond itself and the internalisation of these nanodiamonds cause neurotoxicity? So there's been a whole bunch of different cytotoxicity, biocompatibility studies done with diamond in neuronal systems. But what we wanted to know was, does the presence of the diamond actually impact the function of the neuronal network? Because that's a key question. If you want to use these as a sensor, the sensor can't perturb the function of that network. And so that was the first question we were trying to answer. The second one was, can we implement these quantum sensing protocols? Can we do this ODMR, if you like, on multiple nanodiamonds across wide fields of view simultaneously? So most of the ODMR experiments that have been performed have been done using a confocal microscope where you get access to 1 nanodiamond at a time. So we want to really scale that up. So to do that, we worked with some colleagues over at the Florey Institute of Neuroscience in Steve Petru's group, and we use these multi-electrode arrays. And these arrays, what you're looking at here is these are 24 well plates, and in each one of these wells, we can culture a neuronal network, and there are 12 reporting electrodes in each one of these wells. So we do the culturing in each one of the wells. We then keep some for controls. We then add nanodiamonds to others. And what we want to know is we want to compare the firing dynamics of the control versus the firing dynamics when we add the nanodiamond and post addition of the nanodiamond. And so the nanodiamonds that we used in this work were 100 nanometres in size. They had about 500 NVs per nanodiamond, so they're very bright, and you can track them easily in this wide field imaging system. And here we didn't do any specific functionalisation. This is just with bare nanodiamonds, incubating the nanodiamonds with the neurons and then looking 24 hours after the addition. And so this is what some of the electrical recordings look like. So on the left here, you can see action potential events on a single electrode. So this is over a period of 10 seconds, and these little blips here are an action potential happening on one of these electrodes. Remember, there are 12 electrodes for each one of those wells, and we can monitor that for 24 wells all simultaneously in this system. And you can generate what are called these raster plots. And these raster plots are basically assigning an action potential event. You'll basically get a blip. And then you can look at much longer timescales. So the typical recordings we would do in this work went for 5 minutes. And then using these raster plots, then you can start to analyse whether there's changes in the firing dynamics between a control and when you add the nanodiamond. And so after we added the nanodiamonds, these are what the recordings look like. So just comparing the raster plots, you can't really get a feel for whether there's a change in the dynamic or not. So what you need to do is a more rigorous analysis, and this is what the team at the Neuroscience Institute have been doing. And I haven't got time to go through all of the details here, but basically you can generate what are called Orous plots. And these plots, the way to interpret these plots is that if you see a change in colour away from grey, that change in colour will represent a statistically significant change from the control to when you added the nanodiamond. And these labels on the side are 25 different network parameters that you can compare from the control to after the addition of the nanodiamond. And we've done it for 12, 24 and 36 hours after the incubation period of the nanodiamond. And you can see that there is no statistically significant difference in any of the 25 network parameters that we looked at. And furthermore, you can do the same thing if you vary the nanodiamond concentration. So here we varied it from 1 micromole up to 20 micromole, and we didn't see any statistically significant change. So that says that there is good evidence to suggest that there is no neurotoxicity caused by the presence of the diamond, which is an important result if we want to go on and use these for sensors. In terms of imaging, so if you want to do wide field imaging, so now what we did was cultured these neurons onto our wide field imaging microscope. This is a schematic of the wide field. We've got a microwave resonator, so we can drive the ODMR. The neurons are cultured within that resonator, and then we've got culture media on top to keep them all happy. And so you can use the ODMR, so people have showed this before, you can use ODMR to perform background-free imaging. So the ODMR, you can modulate the intensity of the NV, but you don't modulate the cell autofluorescence. So you can actually remove all of the cell autofluorescence. And then this is what a typical area of a neuronal network looked like. So that you can see this was cultured with 8 micromole of solution. So we get great coverage in terms of the nanodiamonds on these dendrites. And what we did was performed wide field ODMR by simply taking 30 millisecond exposures with different microwave frequencies. And so if you do that, within 3 seconds you can build up this ODMR spectrum. And within 3 seconds we have that ODMR spectrum for every nanodiamond that you see in that image. So this is a considerable scale up compared to confocal when you're just looking at one. And so to demonstrate that we can use this for sensing, this is more of a proof of principle. So we didn't see an intrinsic change in the temperature with these neurons. But what we did was change the environmental conditions from 37 to around 35. And then we tracked the same nanodiamond before and after this temperature change. And the shift in the ODMR is consistent with that change in environment that we introduced. Okay, so that suggests that these probes will be as sufficient or have sufficient sensitivity to probe temperature changes in these sorts of systems. And what can we look at? So recently there was work suggesting that mitochondria can actually function at temperatures as high as 50 degrees. So this is something I think that diamond can really help verify whether this is actually happening or not. And there's also this theoretical work that was put out in the mid-90s which suggests that ion flux through an ion channel, which is a result of an action potential event, may actually cause a temperature increase as well. So if you can get nanodiamonds close enough to these ion channels, then there may be a significant temperature change that we could think about measuring with these sorts of probes. So that's the story from the nanodiamond side of things. The next part of the story is some of the new work that's coming out of the group. And so here Again, this is based on this quantum magnetic microscope that we've developed, and we're currently preparing it for publication. So yeah, I'd ask that you don't share these images if that's okay. And so the idea here is that we want to use, before I go to that, we want to use these single crystal diamonds and this magnetic microscopy to actually answer some interesting questions about biology. For a long time, we've done a lot of proof of principle experiments about what we could do, but we've found recently that there's an interesting system here in this sea mollusk where we can start to answer significant questions. And so this sea mollusk is found all around the world. This is the one that's found in Western Australia, and the one that we've been doing most of our work on. And the interesting thing about these is they spend their life sitting on a rock and they eat rock to extract food. That's what they do all day. So they mine this rock, if you like, and then they extract algae out of that rock, and that's how they function. Now, to do that, they need incredibly hard teeth. And so they mineralise their own teeth by themselves, and they wear through one tooth every 2 or 3 days. And so if you get one of these animals and you extract what's called the radula, the radula is the organ that contains all of their teeth. Okay? And you can see this mineralisation process happening. And the mineralisation, the material that they make their teeth out of is iron. Okay? So they have magnetic teeth. And so you can see here at the beginning where you've got clear teeth, there's no iron present there. And then the teeth start to change colour. So they start to get a little bit orange. And then that represents the point at which it starts to mineralise the iron into a particular form called magnetite. And as these teeth get developed further and further, it's just getting more magnetite, and it's assembled in such a way that this material at the end, which the animal uses to scrape the rock, is the hardest biomineral that people have identified. So it is harder than stainless steel, it's harder than engineered ceramics, and this system, this biological system, is able to do that sitting in seawater at 15 degrees, just sequestering iron from its environment. So it's an amazing system how it's able to assemble this iron mineral in such a way to get these amazing mechanical properties. So chemists and material engineers are interested in reverse engineering this process. And if we can understand how this animal does it, then we can start to synthesise this sort of material, and we can create new advanced functional materials that are lightweight, wear resistant, and super hard. And so that's the motivation behind it. And traditional studies on these teeth have been done using synchrotrons. So typically you would go and cut a section of the teeth, take it to the synchrotron, and you would get X-ray fluorescence images that would look something like this. Now these images tell you about the different mineral components. So for example, down here, the red indicates where there's magnetite, the blue indicates where there's ferrihydrite, and ferrihydrite is the precursor mineral. So the ferrihydrite feeds the mineralisation of magnetite. That's how it works in this system. So this tells you where the elements are, but it doesn't tell you anything about how they're getting assembled. And this is where we want to use the magnetic microscope to tell us a little bit more information about this system. And so as a quantum physicist, you don't get to go on many field trips to source your samples. So I was fortunate enough to start to collaborate with Jeremy Shaw. So he works at the University of Western Australia. He's one of the world experts on iron biomineralisation of these animals. And so we went snorkeling off the coast of WA. We got a couple of these samples. We prepared them for imaging on our microscope. And this is an example of what the radula looks like. So this is a resin-embedded radula, which has been imaged with micro-CT. And so we need to understand what the orientation of the tooth is, and then we want to take a cut along a specific direction so we know what we're looking at. And that's what a single cut looks like. So we typically take a 1 micron slice, and then we put that slice on top of our diamond imaging chip. And then we want to image the magnetic properties. And so this is what we get. So if you put down these teeth, you can see here this clear tooth is before mineralisation. The orange starting to develop here is called the onset of mineralisation. And then you can see there's more magnetite being formed as the tooth get older. And if you do ODMR at every pixel on this wide field camera, you can start to resolve regions where there is magnetite forming. So magnetite is ferrimagnetic, so it provides a static magnetic signal. So it can actually image where the magnetite is in this tooth with much higher resolution than what you would get with a typical synchrotron, for example. So here we're limited by 300 nanometers. And so you can see that when you're on the tooth, you get ODMR that is split, and when you're off the tooth, we get a weaker field. So we can measure the fields coming from these teeth. What's more, we can look at the precursor mineral as well. So I haven't got time to explain what T1 relaxometry is, but there's a different imaging mode which can detect fluctuating magnetic fields, and we do that with what we call T1. And ferrihydrite, which is the precursor mineral, is super paramagnetic, so it has a fluctuating magnetic field. And so we've got two imaging modes. One, we can image the magnetite, and two, we can image this ferrihydrite. And now we get a full elemental map of both important minerals in this tooth, and we can do it over the whole region of the tooth. And what's important here is you want to identify where there's regions of overlap, because where there's overlap, you've got this phase transition from ferrihydrite to magnetite. And so if you want to do high-resolution TEM studies, you need to know where to look instead of just putting it in and taking a guess. So these high-resolution maps provide important information for future studies in that regard. But more importantly, what we want to use the NV for is to do vector magnetometry. So I said before you have these four different orientations of the NV. That means you can measure this magnetic field in these four different directions, and then you can generate a map of the magnetic field in the X direction, the Y direction, and the Z direction with respect to that tooth. And if you've got all of those components, now you can reconstruct the full vector magnetic field. And so that's what it looks like here on this tooth. And so here you're looking at regions where, so now you're looking at the strength of the magnetic field. I don't know why it's not playing there. But, so you're looking at the strength of the magnetic field and the direction as well. And you can see that there's incredibly complex behaviour going on here. And this helps us get to how this system is able to assemble this sort of material. Well, I don't know why it's capped it. Okay, hang on. So if we, hopefully that'll work. Yep. So now if we take a zoom in on these vector profiles, what you can see if you look at the tip of the tooth, this is what the magnetic field is doing. So the magnetic domains at the tip of the tooth are predominantly perpendicular to the edge of that tooth. And as we go down into this blue box here, again we've got perpendicular. And then at the base of the tooth, what you can see is the magnetic profile changes and wraps around. And this change is a structural design of the tooth. This is one of the reasons why it's able to be wear resistant. And so there's been structural studies performed on this teeth on a different species, and that shows exactly the same thing. So this is what a fully mature tooth looks like, where you have this rollover at the base of the tooth, and this is what it looks like in the middle of the tooth. And so this is a structural design that the chitin have evolved to do to make their teeth wear resistant. And what we see is that this is happening at the very onset of mineralisation. And so that means that the way the iron is coming and depositing on this system, it's doing it in an oriented fashion. And that's something that the biologists didn't know before. And now that we have that knowledge, we know that we need to target the organic matrix and how the iron attaches to that matrix to be able to reverse engineer this system. And that's what we're trying to do at the moment with colleagues in the US. So with that, I want to wrap up. So today I've given you a snapshot of some of the opportunities to do with nanodiamonds and single crystal diamond in terms of thermometry, vector magnetic imaging and magnetic profiling of biological systems. There are a whole host of different applications that apply to biology that we're also pursuing. So recently we were able to show that we can hyperpolarize proton spins. And this technology will be important for improving contrast in MRI imaging. So we're hoping to be able to scale this up from a single NV case to the ensemble. We're doing some work with the EPR microscopy, which I mentioned. We're also doing a lot of work with magnetic bead imaging, so we can image magnetic fields from single magnetic beads, watch them, how they evolve in biological systems. Neuronal sensing and interfacing with nanostructured diamond, and a stack of nanoscale MRI and NMR, which again we'll hear more about in the magnetometry session tomorrow. So I think the opportunities for diamond quantum probes for biosensing and imaging is broad. I think we've got a lot of work to do in terms of the materials aspect, so I'm happy to see people are starting to look at the effects of the surface on these quantum properties, and I think that if we can improve these material properties even further, these applications are going to go far beyond what I've talked about today. So with that, I want to thank the organisers again for the very kind invitation, and thanks very much for your attention.